Hello, this is Professor Hudson. You're going to view a narrated PowerPoint lecture on meiosis and sexual life cycles. We're going to continue with Concept 3, which talks about meiosis and how it reduces the chromosome number from diploid to haploid. Then we will talk about genetic variation that occurs during meiosis and how it contributes to evolution. Let us begin. When talking about meiosis, there are three events unique to meiosis that do not occur during mitosis, and all three of these occur in meiosis 1. Synapsis and crossing over in prophase 1. Homologous chromosomes physically connect and exchange genetic information. Homologous pairs line up at the metaphase plate, and then there's separation of the homologs during anaphase 1. In mitosis, the homologs never pair up. They line up single file at the metaphase plate. But during meiosis, particularly meiosis 1, you will have the homologs, or the sets of chromosomes from each parent, pairing up so that they may do synapsis and crossing over, and then they will separate. Meiotic cell division occurs in animal ovaries and testes and is a prerequisite for sexual reproduction in all eukaryotic organisms. Meiotic cell division involves a specialized nuclear division called meiosis and 2 rounds of cytokinesis. 2 divisional steps produce 4 daughter cells that can become gametes. Daughter cells are genetically different from the parent cell and from each other. Each daughter cell has half the genetic material of the parent cell. When this process occurs in the male, we call it spermatogenesis. 4 haploid sperm are produced. When this process occurs in the woman, we call it oogenesis. Note that in reality, the woman only produces 1 usable egg, and the remaining 2 to 3 cells are called polar bodies, whose whole purpose is to get rid of the extra chromosomes or the extra DNA so that the egg produced will be haploid. Also note that in women, meiosis 1 occurs, and then meiosis 2 only partially begins. Meiosis 2 will only finish if the egg is penetrated by a sperm. Let's review. In females, meiosis is part of blank, which occurs in the ovaries and reproduces eggs. Choose the correct answer, and this is question 14. In this figure, there's a comparison of mitosis and meiosis. Notice in mitosis, there's only 1 division and the cells remain diploid. In meiosis, there's 2 divisions and the cells become haploid. Also in meiosis, genetic recombination occurs so that each haploid cell that is produced is slightly different from the others. This does not happen in mitosis. In mitosis, the daughter cells will be identical to the parent cell. In meiosis, you get 4 cells. At the end of mitosis, you get 2 cells. Mitosis plays a role in growth, development, and repair in body cells, and meiosis only occurs to produce haploid sperm and egg. It has no other role in the eukaryotic organism. This figure summarizes some of the differences between mitosis and meiosis. Make sure you're aware of some of the differences and similarities. Question 15. Homologous chromosomes separate during blank, and then the sister chromatids separate during blank. Choose the correct answer. Question 16. Which is true of both meiosis 2 and mitosis? Choose the correct answer. Now we will finish up with our last topic or concept for chapter 10. Genetic variation produced in sexual life cycles contributes to evolution. Genetic variability among organisms is essential for survival in a changing environment. Mutations produce a new variation but are relatively rare occurrences. The genetic variability that occurs from one generation to the next results almost entirely from meiosis and sexual reproduction. This slide lists the different types of genetic recombination that occur in meiosis. Two occur actually during meiosis, and then there's some additional levels of genetic recombination that occur as a result of fertilization. Synapsis of the homologs leads to crossing over of non-sister chromatids at the chiasmata, and independent assortment of homologous chromosomes also leads to genetic variability. The synapsis and crossing over occurs during prophase 1 of meiosis 1. The independent assortment occurs during metaphase 1 of meiosis 1. And then you get additional genetic recombination from the fact that fertilization under natural circumstances is random. Any one of millions and millions of sperm can fertilize any one of thousands and thousands of eggs. This slide is showing an example of synapsis and crossing over. Also, there is an electron microscope picture of an actual set of chromosomes or homologs undergoing synapsis. The red circle is being used to highlight the electron microscope figure showing two chromosomes undergoing crossing over. Synapsis is when they line up close together, and the crossing over is when the DNA is exchanged. The regions where the synapsis and crossing over occurred is called the chiasma or the chiasmata. Synapsis and crossing over is also shown here. Notice that the crossing over is occurring between the chromosome from dad and the chromosome from mom that has the similar genes. This is important. You would not want a gene for eye color from dad recombining with the gene for, say, skin color from mom. When genetic recombination occurs, it needs to occur between the same types of genes. Crossing over creates chromosomes with novel combinations of genes. Because sexual reproduction can bring together homologous chromosomes from two vastly diverse populations, crossing over between these homologs can introduce allelic combinations that are completely new. The resulting gametes are potentially very valuable to the evolution of the organism. Crossing over during prophase 1 of meiosis 1 ensures that no pure maternal or paternal chromosomes exist within humans. So when the chromosomes are passed into the sperm and the egg during the meiosis, the 4 haploid sperm or eggs, while they do contain the DNA from the mother and the father, they have been scrambled a little bit to give unique combinations that may not have been seen in the mother or the father. Crossing over is why you can have 4 siblings in the same family with the same mother and father, and they can look very different from one another. They came from the same parents, however, crossing over, independent assortment, and random fertilization are reasons why each child will look different from the other. Let's watch an example of meiosis with crossing over.
Crossing over during meiosis allows recombination of genes between homologous chromosomes. This alters the linkage between genes on the same chromosome. Crossing over occurs frequently during prophase 1 of meiosis, when homologous chromosomes associate with each other in a structure called a tetrad. First, consider the possible inheritance of alleles for 2 genes on a particular chromosome when crossing over does not occur. Recall that one member of each pair of homologous chromosomes came from one parent. The other member came from the other parent. In this example, one parental chromosome had alleles capital A and capital B, while the other had alleles lowercase a and lowercase b. Without crossing over, each haploid cell produced will inherit a chromosome with the same combination of alleles as was on one of the parental chromosomes. Thus, the arrangement of linked genes is not altered in this case. Now consider the case where there is crossing over in the region between the two genes. Since the crossing over is only between two individual chromatids, the possible haploid cells after meiosis have 4 different genetic combinations. 2 will come from the chromatids that did not participate in crossing over and thus will still have the parental combination of alleles, either capital A and capital B, or lowercase a and lowercase b. The other 2 will be recombinant, with either capital A and lowercase b, or lowercase a and capital B alleles. Thus, crossing over allows for the reassortment of linked genes.
This slide is showing another type of genetic recombination that occurs called random assortment. When the homologs line up at the metaphase plate in metaphase 1, they can line up in different combinations or in different parts of the metaphase plate as long as they're next to their partner. This also gives genetic variability because when the cell divides in half, the different arrangements of the chromosomes will dictate which cells get which members of the homologous pair. Let's take a look at an animation on random assortment.
During metaphase 1 of meiosis, each joined pair of homologs lines up on the metaphase plate. The orientation of each pair on the spindle axis is random. Either the maternal or paternal homolog may orient toward a given pole. The number of possible chromosome orientations equals 2 raised to the power of the number of chromosome pairs. In this hypothetical cell with 3 chromosome pairs, 8 2 to the third possible orientations exist. Each orientation produces gametes with different combinations of parental chromosomes.
Outside of crossing over and random assortments, which occurs during meiosis, additional variation occurs when any one sperm can get with any one egg. Fusion of gametes adds further genetic variability to the offspring. 2 gametes during fertilization, each containing a unique combination of alleles, fuse to form a diploid offspring. Each gamete, by independent assortment alone, can have 2 to the nth possible combinations. Fusion of gametes from 2 individuals can create 2 to the n times 2 to the n possible combinations. Therefore, gametes from 2 humans could produce about 64 trillion different combinations. This means there are 64 trillion possible ways your children can come out each pregnancy. Question 17. The combining of chromosomes from genetically different gametes occurs during. Choose the correct answer.